Um, Hello, dear students, my dear future doctors. Um, this is Dr. Madonna speaking to you. Um, Welcome to the Fundamentals of Medicine. I'm going to be your lecturer from today on in this sequence of lectures. So it's almost 1 o'clock, so I'm going to start recording this lecture. I hope you're going to join me by the time I start actually talking about the subject of today's material. So before that, I would like to remind you what it is about. Okay, what's genetics, or what kind of role it plays in medicine, and so on. Okay. So meanwhile, I'll just start sharing my screen, and I hope the rest of you can join me as well. So this is my first lecture in this new subject, so just bear with me. I take all your suggestions and so on. Okay. So let's start with the general terms. Okay. In order to understand how important genetics is in medicine, we should understand the nature of the hereditary material, okay? How it's packaged into the human genome and how it's transmitted from one cell to another, and how it's divided into many cells from one cell, how they become multi-cellular organism, and how the genetic material is transferred from one generation to another generation during reproduction. Okay. The human genome is a huge library of the genetic material that consists of the large amounts of the DNAs, and DNAs are the structural elements, structures of the genetic information. Okay, they carry the genetic information within their structure. Okay, so and also the DNA defines and specifies all the vital processes that undergo and develop in our organs. For example, embryogenesis, development itself, growth and metabolism, and reproduction. Okay, so these are all essential aspects of the human being, right? And this functional unit that transfers this genetic information is the DNA. Okay. Every nucleated cell in the body carries its own copy of the human genome, which, as I said, it's a large library, contains about 25,000 genes, and these genes, which at some point of development define how the genetic information is transferred and how the organs will function, okay, as a union. So, okay, now let's talk about cytogenetics. So, what is cytogenetics? This is actually the oldest branch of the medicine. Back in 1956, this name was called. So, this is the study of chromosomes and their structures and their inheritance as well. So, this is what the cytogenetics is called, and is as ancient as the number of chromosomes defined as 46 in human body. Okay, so this is all I wanted to say as introduction. Now let's go continue with today's topic. So today we're going to talk about that. This is every at the beginning of every my presentation. You're going to see the topic here, the material, the topics titles, and you're going to find the readings. Okay, the chapter of the books you have already uploaded on the drive. Okay. So today we're going to talk about genetics and genomics in medicine, as well as the human genome and chromosomal basis of hereditary material. Also, we're going to talk about cell division and mitosis and meiosis. Okay. So let's start then. Okay, with my first picture here, let's make like this. Okay, so before we look into this picture, let's understand that okay, the genetics is important for the clinical diagnosis of any disease, right? So medical disorders, including Down syndrome, for example, genetic disorders, or microcephaly, they are all because of the visible changes in the chromosome number, either in the case of the Down syndrome or in the structure, and require chromosome or genome analysis, which is possible just by the genetic counseling. Okay, and also gene mapping and gene identification is really important to identify whether the person has the risk of development of this inherited disease or not. And also in cancer cytogenetics, genomic and the chromosomal changes in the somatic cells are often involved in the initiation and also as well progression of the main types of the cells. So you can understand that in oncology, genetics is also important. And also to diagnose many prenatal disease and to calculate the risk of developing certain genetic feature or genetic disease at all. So that is why it's important. So we said that the human genome, I said that, human genome is consists of 46 chromosomes, and it means 23 pairs. From this 23, 22 are actually autosomes. It means the same in both gender. Now it doesn't matter female or male, and just one pair of chromosomes is different between female and male. So this female contains two X's, and the other contains the man contains X, just one X, and the other Y chromosome, right? So you can just say within the human race, we've got the gender differences that is also based on the chromosome at the chromosomal level, the differences between the constitution. Okay, so here we see, if we imagine ourselves the geneticists, what would be the most intriguing thing to do, right? It would be definitely to make a clone of somebody, of a living organism. So here is the first result of the cloning. Dolly, the most famous ship ever existed in the world. And you can see here the Dolly had three mothers, because you can see here 3 DNA particles injected in the egg. Okay, it came from backface Eve, from front-forwarded Eve, and as a result, we've got the surrogate ship. We've got the Dolly, which was the first cloned mammal. Okay. So now let's talk about the cells and the chromosomes. So we know we're dividing organs into prokaryotes and eukaryotes. So we're going to see how they differ from each other. But in both prokaryotes and eukaryotes, the genetic material is organized into chromosomes. This beautiful purple thing, not actually purple, but this is how it looks like for the shape. Okay, and the DNA, each limb, I would say, each part of the chromosome contains the DNA. It's actually made of the DNA, and we're going to see its structure in detail. All life has a cellular base. So let's define what this cellular base means. Okay. Each cell is complicated assemblage of the molecules that can acquire materials, recruit and store energy, and carry out diverse activities including reproduction. So the reproduction is important, the most important feature that leads to transfer information from one generation to another. Okay, the cellular environment. So we said that all the organisms are consisted of cells, not to mention viruses, which are acellular ones, and are capable only to live in other bodies. Okay, for example, coronavirus. The reason we're meeting through this size of the desk. Okay, so living cells are made of many different kinds of molecules. The most abundant part of it is water. So we've got cells and the molecules that are divided in how they feel about water. We've got hydrophilic, which love the water, it means the water soluble ones, and hydrophobic, it means a water insoluble. Okay, they're afraid of water phobic. And the inside of the cell, called cytoplasm, contains both hydrophilic as well as hydrophobic substances. So the molecules that make up cells are diverse in the structure as well as in function. Carbohydrates, for example, they are building materials, glucose, oligosaccharides, and saccharides, as for example starch and glycogen, okay, store chemical energy for the work within the cells, okay. Also, we've got the lipids that also make part of the cells, and proteins, the most diverse molecules within the cell. That are products of the genes. Okay, it means the DNA during transcription. We've got we're writing information into the language of the messenger RNA. From messenger RNA, after translation, we've got the polypeptide chain, which is the protein itself. So the product of the genetic information transfer is protein. And then we've got enzymes that catalyze chemical reactions, different enzymes for different reactions, okay? And this is how the eukaryotic cell looks like. So we've got cells are surrounded by a thin layer called the membrane and specialized structures called organelles. So you can see the shell or the membrane and the organelles within it. This is, again, I repeat, a eukaryotic cell because it contains endoplasmic reticulum, and you can see that the main difference between prokaryote and eukaryote is the nucleus. Okay, nucleus. The genetic material DNA are not free in the cytoplasm, but nucleated in nucleus. Okay, got Golgi, but they've got mitochondria and chloroplast here. So now prokaryotic and eukaryotic cells. So prokaryotic cells are usually less than 1,000th of a millimeter long, and they typically lack a complicated system of internal membranes and membranous organelles as well. The hereditary material that will be here, there is a DNA. It is not isolated in the special cellular conference nucleus. So we've got DNA free lying into the cytosol. Okay, we've got cell wall, they got cell wall, and ribosomes as well, because ribosomes are essential for the protein synthesis. Okay, ribosomes are the organelles on which the protein synthesis takes place. Okay, so eukaryotic cells are larger than prokaryotic cells, obviously, because they contain many organelles that prokaryotic does not. And or they've got one or more mitochondria, which is the source of energy, as you know. The energy recreating organelle also called chloroplast, and some of the plants. In the first slide, we said that it has green particles, also called chloroplast. Hereditary material is contained within a large membrane-bound structure called nucleus, and that is the major difference between eukaryote and prokaryote. Okay? The nuclei of eukaryotic cells provide a safe haven for the DNA, which is organized into, here we've got discrete structures called chromosomes. Okay, and this is the nuclei and DNA within it. This is the cytoplasm. We've got here ribosomes, endoplasmic reticulum, plasma membrane, Golgi apparatus here. We've got centrioles. We've got ribosomes here, smooth endoplasmic reticulum, and lysosomes. Okay. Now let's talk about ribosomes. Okay, we said that they are the organs which are located in the cytoplasm and take a major role in the protein synthesis because they are the place where the protein synthesis carries on. Okay, is carried on. Small organisms involved in the synthesis of the proteins found throughout the cytoplasm, although ribosomes are not composed of the membrane. In eukaryotic cells, they are often associated with a system of the membranous called the endoplasmic reticulum. So this you'll hear my voice better. Okay. Okay. The reticulum may be connected to the Golgi complex, a set of membranous, you can see here in the plasmic reticulum, and you can see the Golgi apparatus or Golgi complex, right? That vesicle, the set of membrane sets and the vesicles as well that are involved in the chemical modification and the transport of the substances within the cell. Okay, you can see here nucleus, you can see reticular at the endoplasmic reticulum, and smooth and the rough one, and ribosomes in here, and Golgi apparatus in here. Okay, so this is how it's called you. The plasma reticulum, smooth and looks like, and this is ribosomes, which are made of two basic subunits. The bigger subunit, large subunit, which is called 50S, and the small subunit, which is called 30S. And here the messenger RNA is located and the translational process takes place. It means that the information from the DNA or nucleotide language, it is rewritten or translated into polypeptide language, and we've got as a result polypeptide chain for the protein. Okay, let me mess it. Okay. And I said that we mentioned there are lysosomes. They are produced by the Golgi complex. These organelles contain different kinds of digestive enzymes that will harm the cell if they were released into the cytoplasm. Both, sorry for that. Sorry. This is from work. I cannot read it on the thread. Both plant and animal cells contain peroxisomes, which are small organelles dedicated to the metabolism of the substances such as fats and amino acids. Okay, and here what makes all the cytoskeleton. Okay, the shape and the activities of eukaryotic cells are influenced by the system of filaments, fibers, and associated molecules that collectively form the cytoskeleton. Okay, these materials give form to cells. Okay, let's do like this. Cells enable some types of the cells to move through the environment, a phenomenon called motility. Okay, they're able to move throughout the cytoskeleton. Okay, they've got the ability of motility. Okay, the cytoskeleton holds organelles in a place, and it plays a major role in moving materials to specific location, because we said that DNA is located in the nucleus, right? And messenger RNA should be transported into the cytoplasm in order to start the process of translation. So, okay, it should leave the nucleus, transported to the site of the protein synthesis to the ribosomes, right? And also, the cytoskeleton takes a major role in moving materials to specific locations within the cells. It's called trafficking. Okay, that's quite a traffic in the cell. And the human genome, you can see here what we're talking about at the beginning. The human genome consists of the 23 pairs of chromosomes, totally 46 chromosomes, and you can see that 22 pairs are autosomes. It means they are same, almost same, okay? But one pair is different. This is X and this is Y. This is what differs the female from male, okay? Females have a double X, while males have just one X and the other Y, okay? It means that a mother can only because the mother's chromosome consisted of just X2X, can only inherit, a girl can only inherit X from the mother, and a boy can inherit also X from the mother. Okay, X are the only thing that mother can give to their both kids. Okay, male and female. While father is able to give a daughter X and the son Y. Okay, so this is how Y is always inherited from the father. Okay, and also given to the son. Okay, and X is always inherited. Either from mother, either from father, but always given to the daughter. Okay, daughter from father, but from mother it can be given to both son and daughter as well. So confusing, but really easy. When you read it, it's gonna be more understandable. The genome contained in the nucleus of the human somatic cell consists of 46, as I said, 23 pairs. 22 are called autosomes, and the other are X and Y are called sex chromosomes. Okay, and here we've got gene location, so it means at any specific locus. Locus is where the gene is located in the chromosome. They may have either identical or the slightly different forms on the same gene is called allos. So you can see two chromosomes, and the allos here are called locuses, which are identical or slightly different. Okay, they're called allos. This one I said, and now let's see the chromosomes more closely. Let's discuss them. This is how it looks like. The chain of the DNA as a chromosome link. Okay, so the chromosome are the structures where the genetic material is located, right? Each chromosome consists of one double-stranded DNA molecule, you can see here, plus assortment of proteins. There are two kinds of proteins, histons and non-histons, and we will discuss them one by one. RNA will also be associated with the chromosomes, and the prokaryotic cells typically contain only one chromosome, although sometimes they also possess many smaller DNA molecules called plasmids. This is for prokaryotes, okay? Now, what are the functions of the genetic material? First, the major function is the genetic material should replicate. It means it should have own the ability to multiply, okay, to reproduce in order to transfer from generation to generation, okay? Because if it does not replicate, it can die within the generation, okay, within one single generation. Control the growth and development of the organs, okay? It directs the process of evolution, directs the process of development of the system, organogenesis, embryogenesis. It's the direct everything, okay? Allow the organs to adapt to the changes in the environment. So, mutations within the genetic material are the essential part of the evolution itself. So evolution combines in itself the modifications that are based on genetic changes, and those modifications allow the organism or an individual to function better in the changed environment. Okay. Yeah, the first man though called them Merkelman, and they came out to the idea in 19th century that the genes carry genetic information and transmit it from generation to generation. So there is a strong evidence that the genes are usually located on the chromosomes. Okay, why not on other structural organelles that we've discussed so far? Why chromosomes? Chromosomes are composed of two types of large organic molecules, which are called macromolecules. These are proteins, and composed of nucleic acids, which can be either DNA or RNA. Okay, so nucleic acids can be deoxyribonucleic acid, and ribonucleic acid can be RNA. Okay, the results of the elegant experiments clearly established that the genetic information is stored in nucleic acids, not in proteins. Okay, nucleic acids, remember. So the proof that why they're stored in DNA, because most of organs genetic information is decoded in the DNA, but in some viruses, as I said, in corona cases, this is the RNA based virus, that is the genetic material is located in the RNA. Okay. Most of the DNA of the cells are located in the chromosome, as I said before, and whereas the RNA proteins are also abundant in the cytosol. The chromosomes and the DNA are located in the nucleus within the eukaryotic cell, or are in the cytosol when we talk about prokaryotic cells. Okay, correlation exists between the amount of the DNA per cell and the number of sets of chromosomes per cell. Okay. Now let's talk about the structure of the DNA and RNA. So with that DNA is usually double strand. What differs from when the RNA, where RNA is a single strand, and it contains four basic nucleotides: adenine, always paired with thymine, and guanine, always paired with cytosine. While RNA is a single-stranded, and this pairing is different. Adenine is instead of thymine, thymine, okay, is replaced with uracil. So adenine instead of thymine is paired with uracil. So AU will got in here in the RNA. This is the nucleotide, their structure. They're composed of the phosphate group, pentose, which means 5 carbon sugar, and nitrogen base group, which is also nitrogen containing compound. As I said, with the human genomic let's say, experiments, they came to conclusion that the amount of the purines and pure mutants are equal. It means that adenine is always adenine plus guanine is always equal to thymine plus cytosine. Okay, here adenine and guanine are double ring bases called purines, what I meant, and cytosine, thymine, and uracil are pyrimidines. And these are the four major players that play the crucial role in the explaining and breaking down the structure of the DNA double helix. Okay, this is Watson and Crick. This is also Francis here, Franklin here, and Wilkins. Okay, they published single-page paper that got them the Nobel Prize. Okay, and paper was about double helix of the DNA, and this is how it looks like, the structural representation. So the amount of thymine is always equal to adenine, cytosine is always the guanine, and the sum amount of the purines is always equal to pyrimidines. Okay. So this is our beloved DNA. Exists in the right-handed double helix, in which the two nucleotide chains are coiled about one another in a spiral. Okay. Sequences of nucleotides linked together by phosphodiester bonds. You can see here. And we've got hydrogen bonds. The two polynucleotide strands are held together in the helical configuration by the hydrogen bonds. Okay, I love to show this kind of thing for you to be easier to understand. So we've got two linear things, and if we do like this, we've got the DNA. Okay, wow, this is this is the DNA, winded and okay, unwinded and winded DNA. Okay, so paradigm once more. Okay, so two strands of the DNA double helix are said to be complementary. Okay, because they gain pairing of the nucleotides. Property of complementarity, two strands of double helix made to be uniquely suited to store and transmit genetic information from generation to generation. The two complementary strands are anti-parallel. Anti-parallel. One starts with 5' and the other starts with 3', and with 3 and 5 consequently. The planar sides of the bases are relatively non-polar and hydrophobic, and this gives the DNA more stable configuration. Okay, that is why it is important to store the genetic material into the stable structure, not like RNA, which is easily broken or unstable, fairly unstable. Okay. So this is DNA we said, and the types of DNA. This is just for your reading. DNA is left-handed, the negative supercoil, and here we've got right-handed positive supercoil, and so on. This is you're gonna find it in your book, in your textbook, and you can read it. Okay. Now let's talk about the diploid state. As I said that every cell contains two pairs of the two copies of each chromosome, right? Many they possess two copies of each chromosome. Okay, 22, 46. Okay, this condition is referred as a diploid state. Okay, it's characteristic of the cells in the body of eukaryote, that is somatic cells, because somatic cells are always diploid. They contain two copies of each chromosome. By contrast, the sex cells of the gametes usually possess only one copy of each chromosome, so they are haploid. Okay, this is called as a haploid condition, haploid state. Gametes are produced from diploid cells located in the germline, which is the reproductive tissue of have organs. Germline cell, I will put that cell, okay? Chromosome structure. It discussed them separately in prokaryotes and in some viruses and in eukaryotes. What chromosome structure looks like. So, DNA molecules of the prokaryotes and viruses are organized into negatively supercoiled domains and monocopies. As I said, just one copy. They have only one set of genes, one copy of the whole gene, right? And genome of the also bacteriophages also like this, and scholars the viral DNA contains 911 genes. They are called folded gene, okay? Bacterial chromosomes that have folded gene. While eukaryotes, as I said, they contain huge molecules of DNA and are highly condensed during mitosis and meiosis, which we can discuss at the end of our lecture, okay? So the centromeres and the telomeres of eukaryotic chromosomes have the unique structure. Centromere means this centromere means the center part, the most condensed part of the chromosome, and the telomeres, which are the four long leaves of the chromosomes. Okay, most eukaryotes are diploid, as I said, with two complete sets of the genes, one each from each parent. Okay, one from each parent. So not only do most eukaryotes contain many times the amount of DNA than prokaryotes, but also DNA is packaged in the several chromosomes, and each chromosome is represented as diploid or polyploid. Chromatin is the complex of the DNA, so chromosomal proteins and some of the constituents of the chromosomes from nucleus. Okay, DNA plus the proteins plus some other components make up a chromosome. Okay, and proteins, which I said, there are two major groups of the proteins: histones and non-histones. Histones are basic positively charged ones that are usually 4 or 5 types of the histones where you have here H1, H2, A2, B3, and 4. Not okay, 4, and they are especially complexed with the DNA to produce the basic structural subunit called the chromatin and the small ellipsoid base called nucleotide. Okay? Histones for nucleosomes with DNA. And we've got non-histones that they are negatively large acidic, negatively charged group of proteins. Okay? And let's say one large DNA molecule contains one chromosome contains one large DNA molecule. The said DNA is the structural component of the chromosome. Okay? Considerable evidence indicates that each chromosome contains a single giant molecule of the DNA that extends from one end through the centromere all the way to the other end of the chromosome. And this is how it looks like. This is the chromatin fiber here. This is the centromere. This is the telomere here. So we've got 3 levels of packaging, DNA packaging in the chromosomes. We've got the first level condensation into negative supercoil, that into the 11 nanometer diameter chromatin fibers, and we've got 13 nanometer chromatin fibers, and lastly we've got tightly packed chromosome, as you can see here on this illustration. And here are the condensation process of the DNA, okay, which happens during meiosis. Here we've got chromosome replication, meiosis, and here we've got multiplication, meiosis 2, which is one strand replicated. Okay, we've got 4 haploid gametes as the result. Okay. So now let's talk about centromeres and telomeres. I mentioned them briefly, but I didn't describe what my way. So the centromere of the metaphase chromosome can usually be recognized as a constricted region, presence of the specific DNA sequences that are repeated many times frequently in the long tandem areas. Each chromosome of the human, these are telomeres, okay, the ends of the chromosomes. So each centromere of the human chromosomes, for example, contains 5,000 to 50,000 copies of the satellite sequences, which are also located at the chromosomes. And telomeres, they are the ends of the eukaryotic chromosomes that have unique properties, and we've got the unique enzymes called telomerases. Their major function is to protect telomeres and keep them as long as they are, okay? Keep them from shortening age by age, because telomeres shorten after every cycle of reproduction. Natural ends of the normal and broken chromosomes are stable and show no tendency to fuse into another DNA of the broken or native ends. Okay. And about telomeres, one more, they prevent DNA from degrading the ends of the DNA molecule ribonucleases, or the enzymes that break them down. They prevent fusion of bands of the different DNA and mixing up the genetic material, and facilitate the replication of the ends of the linear DNA without the loss of material. Okay. As the age increases, the telomere size decreases. So it means the telomere is somehow linked with the aging. Okay, and if there is an active telomerase, it protects the telomerase, which means the telomerase acts as an anti-aging factor. Okay. In normal, non-cancerous human somatic cells, telomeres usually contain 500 to 3,000 repeating sequences and gradually shorten with age. In contrast, the telomeres of germline cells and the cancer cells do not shorten with age, okay? Because they have the uncontrolled ability to multiply and replicate themselves. So it means it should be definitely somehow linked with the telomerase length, okay? Because they have still legs and they don't extinct, they don't die out because they multiply uncontrollably, unstoppably, okay? The telomeres of the humans and a few other species have been shown to form structure called T-loops or loops generally, in which the single strand at the 3' terminus invades the upstream telomerase repeat and pairs with a complementary strand, displacing the equivalent strand. Okay, T-loop, that is what it is. The DNA in the T-loops is protected from degradation or modification by DNA repair process telomerase specific protein called shelter. Shelter is somehow gives a shelter to this T-loops, okay, to protect the T-loops from degradation. The chromosomes of eukaryotes contain many DNA sequences that are repeated in the haploid chromosome complement, sometimes as many as 1,000,000 times. DNA containing such repeated sequences is called repetitive DNA. It's a major component of the eukaryotic genes. And this is the end of the first part of the lecture. Then we're going to discuss cell division, but before that, let's summarize the things. So, okay, now that we've got satellite bands, we talked about it, we talked about denaturation and renaturation, which is okay, DNA molecules. If they're heated at the 100 Celsius degree, they bond between the DNA, hydrogen and phosphatide bonds. They break down and they denaturate. Okay, when the complementary strengths are separated, it means they are denaturated or denaturation. But if we provide them with the proper conditions, they can renaturate itself. It means they can form double helix again, okay, if we provide with the proper conditions again. A key point of all this is that the eukaryotic chromosome contains one giant molecule packed in the 11 nanometer nucleosomes. We've got the second stage of condensation. We've got 30 nanometer chromatin fibers that are present during mitosis and meiosis. Okay, and metaphors, the 30 nanometer fibers are segregated into domains by scaffolds composed of non-histone chromosomal protease. And lastly, the centromere and telomeres, which is centromere the spindle fiber attachment regions, and the telomeres termini of the chromosomes, which have unique structure and this facilitates the function of the chromosomes. Okay, and the unicity or the specificity of the cell. And eukaryotic genomes contain repeat DNA sequences with some gene sequences present a million times or more. And the repeating sequences also might lead to some genetic disorders like Huntington's disease. This is the sequence of repeating the disease with and the frequency of this repeating sequence. It defines the onset of the disease as well as the severity. Okay. So, okay, let's have 10-minute break, and I will be back with the presentation. Okay, let's have 10. Oh, I'm just all by myself here, so I'll have a break. If you don't mind, and you cannot mind because you're not here. See you in 10 minutes then. Okay, let's go on discussing the cell division then. Just give me a second, I will share my screen with you. Okay, I think we're good. Okay then, so we said that in order to pass the genetic information from one generation to the other, it's important that they replicate, okay, or reproduce. Cell division plays a key role in the transfer of genetic information, right? The cell can divide into two cells, each of which can also divide into two, and so on and so forth, and as a result, they create a population of the cells called clone. Okay? The cell that is about to divide is called mother cell, and the product of the division is called daughter cells. Okay, from one mother cell we've got two daughter cells. When prokaryotic cells divide, the contents of the mother cell are more or less equally apportioned or how to divided in between of the two daughter cells. Okay, this process is called fission. So the proportion division of the genetic material between two daughter cells is called fission. This is what the cell division looks like, mitosis and meiosis. Okay, we'll discuss them one by one. Let's start with mitosis. Each time a eukaryotic cell divides, it goes through the series of phases that collectively form the cell cycle. Okay, the living cycle. The progression of the phases is denoted like G1, S, then G2, and M. S in this means DNA synthesis. Okay, it's a period in which the chromosomes are duplicated, or DNA synthesis, and S from the synthesis. Okay, while M refers to the term mitosis itself. So phase in the cell cycle and the time when the mother cell actually divides, not just the DNA synthesis, but actual division of the cell. Okay. These phases usually have two components. The first will be mitosis itself, and the second is going to be cytokinesis. And let's discuss one by one. So this is interphase, G2, prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. So this was all mitosis. And lastly, we've got cytokinesis. So let's say what was mitosis. This is the process that distributes the duplicated chromosomes equally and exactly to the daughter cells, while cytokinesis is a process that physically separates the two daughter cells from each other. Okay, mitosis is duplicated chromosomes to equally be distributed in the daughter cells, while cytokinesis is the direct physical separation of these two daughter cells. Okay, the G and the G2 phases refer as gaps. Okay, the gaps between synthesis and mitosis. Okay. Here is what it looks like. Stages of mitosis: interphase, prophase, late prophase, metaphase. This is all the same. Metaphase. Remember, the center located, and then anaphase, fusing off, and telophase, lastly, and cytokinesis, the final physical separation of two daughter cells. The key points of all this: cells, the basic units of all organisms, they are enclosed by membranes, as I said. The chromosomes, the cellular structure, carry these genes. They're composed of DNA, RNA, protein. And eukaryotic chromosomes are contained within the membrane-bounded nucleus, and eukaryotes they are not. And the eukaryotes, they possess systems. Okay, I think this one will be better before if we say this one before the subdivision. Okay. Okay, so they are both like this. They possess each copy of the each chromosome, one copy of each chromosome, And the prokaryote cells divide by fission, as I said. Eukaryotic cells divide by mitosis and cytokinesis. And the eukaryotic chromosomes duplicate when the cell's DNA is synthesized, and this event, which precedes mitosis, characteristic of the S phase. Okay, so let's go on then with the phases. Okay, with mitosis. The eukaryotics, as I said, they are divided in eukaryotic cells. The distribution of the genetic material is almost equal, that is by means of fission. Okay, so the genetic material equally and exactly is transferred to their offspring. The network of thin strands formed by all the chromosomes within the nucleus is referred as chromatin, as we said. And during mitosis, the chromosomes shorten and thicken. This is called condensation. Also they can decondensate. It means that after mitosis, the chromatin network is reformed. Okay? And biologists often refer to the period when individual chromosomes cannot be as interphase, okay, cannot be seen, okay, at all. This period, which may be quite lengthy, and the time between successive mitotic events, okay, interphase, in between phase, not seen chromosomes, not visible chromosomes. When mitosis begins, each chromosome has already been duplicated. Okay, mitosis begins, but chromosome already we've got two pairs of them. Okay, the duplicates called sister chromosomes remain ultimately associated with each other and are joined at the chromosome centromere. We said centromere is the central part, the most condensed part. The distribution of the duplicated chromosomes to the daughter cells is organized and executed by microtubules, which are components of the cytoskeleton, as we said. During mitosis, the microtubules assemble into the complex array called the spindle. Okay, the formation of the spindle is associated with microtubule organizing centers or MPOCs. Which are found in the cytoplasm of the eukaryotic cell, usually near the nucleus. Okay? In animal cells, the mitosis are differentiated into small organelles. They are called centromeres. Okay? And these organelles are not present in the plant cells. Okay? They are not present in the plant cell, just in the animal and human cells. The final positions of the centromeres define the roles of the dividing mother cells. The initiation of the spindle formation, the condensation of the duplicated chromosomes from diffuse network of the chromatin, all hormones of the first stage of mitosis called prophase. Remember, first stage, prophase, alignment of the spindle and the chromosomes. Formation of the spindle is accompanied by fragmentation of many intracellular organs, for instance, the endoplasmic reticulum and Golgi complex, and nucleolus, a dense body involved in the RNA synthesis within the nucleus, also disappears during prophase. Mitochondria and chloroplasts remain intact. They do not take part into the formation of the spindles, okay? And again, the concomitant with the fragmentation of the plasmic reticulum, the nuclear membrane breaks up into many small vesicles, and microtubules form within the cytoplasm, invade the nuclear space. Some of these microtubules attach to kinetochores, which are called protein structures associated with the centromeres on the duplicated chromosomes. Attachment of the spindle microtubules to the kinetochores indicate that the cell is entering the metaphase of the mitosis, or the second phase of the mitosis. Prophase, then attachment to the kinetochores of the spindle, will called metaphase. Okay, and it is seems complex when I'm reading, but when you're gonna see illustration, it it will make sense, okay? Through the operation of the spindle apparatus, the duplicated chromosomes come to lie in a single plane in the middle of the cell. So this is what metaphase looks like. This equatorial plane is called metaphase plate, okay? And the sister chromosomes with the duplicated chromosomes are separated from each other during anaphase of mitosis. We get prophase, we've got metaphase, and then anaphase, okay? The separation is accomplished by shortening microtubules attached to the kinetochores and by degrading materials that hold the sister chromatids together. The decondensation of the chromosomes and the restoration of the internal organs are characteristic of telophase of the mitosis. Telophase is the regeneration of the first first phase of this cell. Okay, when mitosis is complete, the two daughter cells are separated by the formation of the membranes between them. Okay, they've already got their own organelles, they've got already their chromosomes, and they are separated within their own membranes. Okay. So the cell enters mitosis as duplicated chromosomes condense into rod-shaped bodies. This happens everything in the prophase. This is the summary of mitosis. So prophase, we've got the duplicated chromosomes condense into rod-shaped bodies. And during metaphase, chromosomes migrate to the equatorial plane of the cell or metaphase plate. And then during anaphase, the centromere that holds the sister chromatids and duplicated chromosomes split, and then also microtubules shorten and the chromatids separate, okay, from each other. This happens in anaphase. And lastly, it's telophase. The mitosis comes to its end, the chromosomes decondense, and the nuclear membrane forms around them as well. The cell membrane reforms around the or cytoplasm and organelles. Okay, each daughter cell produced by mitosis and cytokinesis has the same set of chromosomes that the daughter. Okay, daughters are genetically identical cells or clones. So this was all about mitosis, and now let's talk about meiosis. Okay, this is quite simple. Mitosis is the simplest way of replication. Okay, producing two output out of one, and meiosis, this is a sexual reproduction, involves a mechanism that reduces the number of chromosomes by half. Okay, in case of mitosis, we had already the DNA, the chromosome, the amount of chromosomes were always duplicated, okay? And they just separated. And in meiosis, we've got the reduction of the genetic material into halves. Meiosis, this is from Greek word meaning diminution. It is a process that reduces the diploid state to the haploid state. That is, it reduces the number of chromosomes in the cell by half. The resulting haploid cells either directly become gametes or divide to produce cells that later become gametes or germ-like cells. Okay? Meiosis therefore plays a key role in reproduction among eukaryotes. This is how meiosis looks like. Okay? Each pair is distinct. Different pairs of the chromosomes carry different sets of genes. You can see here this is the gene interface during meiosis, and each pair as a result uploaded includes the different, each carry different types of the sets of genes. The members of a pair are called homologous chromosomes or simply homologous from a Greek which means agreement. Okay? Homologous carry the same sex of genes, they may carry different alleles of these genes, okay? And chromosomes for different pairs are called heterologous, okay? This one is done, and all of them are heterologous, okay? So the process of meiosis involves two cell divisions. We've got meiosis 1 and we've got meiosis 2, which we didn't have during mitosis. Chromosome duplication, which is associated with the DNA synthesis, occurs prior to the first of these divisions, okay? It doesn't occur between the two divisions, okay? Prior to the first. The progression of events is that we've got chromosome duplication, we've got meiotic division 1, and we've got meiotic division 2. So then in between, there is no chromosome duplication, so as a result, we've got half of the chromosomes, right? If we represent the haploid amount of DNA by the letter C, then in the sequence, the eventual double amount of the DNA is called 2C to 4C. So cut in half from 4C to 4C. Finally, we've got C as a half. Okay. The overall effect is to reduce the diploid chromosome number, which is diploid 2N, to the haploid, means single N. Okay. This is the stages of the meiosis. Meiosis 1, meiosis 2. The prophase again, we've got metaphase, slightly different, anaphase and telophase. So the sequence is the same, but the meaning is changed. Okay. So the prophase of meiosis 1, or simple prophase 1, it's denoted by the Greek term. This term conveys key features about the appearance of behavior of the chromosomes. So leptonema from Greek words meaning that thin threads is the earliest stage of the prophase 1. During leptonema, the duplicated chromosomes condense out of the diffuse chromatin network. Okay? The chromosome condensation continues, the cell progresses into zygonema, leptonema into zygonema, and during zygonema, homologic chromosomes come together intimately, and this process of appearing between homologous is called synapsis. Okay, remember synapsis. Leptonema, zygonema, synapsis. Okay. Here we are again at the meiosis 1. The structure called the synaptonemal complex, okay, consists of 3 parallel rows, one associated with each other of the chromosome called lateral element, and one located midway between them. It's called central element. And a large number of ladder-like rounds connected the lateral elements with the central ones. The role of this synaptonemal complex in chromosome pairing and in subsequent meiotic events is not fully understood, but it is known that it's an essential part of the first phase of the meiosis. Okay, here we've got replication and recombination, we've got cell division, and then starts the meiosis. Until here we're discussing that. The thickened chromosomes that result from the process are characteristic of the pachynemma. Pachynemma is different. It's a thick thread. Thick. A pachynemma of paired chromosome can easily be seen with a light microscope. Okay, they're visible with the light microscope. Each pair consists of 2 duplicated homologies, which themselves consist of 2 sister chromatids. If we count homologies, a pair is referred to as bivalent. B means 2 bivalent chromosomes or bivalent chromosomes, whereas if we count strands, this is referred as tetrad of chromatids. Tetrad means 4 chromatids. Okay, bivalent chromosomes and tetrad of chromatids. During pachynemma, or perhaps a bit before or after, the pair chromosomes may exchange material, and this process is called, the phenomenon is called crossing over. The change in between with materials is called change over. And diplonym, during diplonema, the pair chromosome separates slightly. However, they remain in close contact where they have crossed over. So these contact points are called chiasmata. Okay, it means cross. So the point where they crossed as they cross over the information are called chiasmata. They remain close to each other. The close examination of the chiasmata indicate that each of them involves only 2 to 4 chromatids in the tetrad. The deflutant stage may last a very long time. In human females, for example, it exists for more than 40 years. This is meiosis 1. This is an illustration from the Thompson books. Also, you can use principles of genetics. So you are not subsided with, or you're not constricted to use of one or the other. You can use both of the books, or you can choose which one you prefer. Okay, principles of genetics or Thompson's like that. Okay. The chromosomes still held together by the chiasmata then move to the central plane, which is again metaphase, right? It's perpendicular to the axis of the spindle apparatus. This movement is characteristic of the last stage of the prophase 1 and called diakinesis, means movement through. And there is when metaphase 1 begins. You can see here the pair chromosomes orient toward opposite poles of the spindle. And the orientation ensures that when the cell divides, one member of each pair will go to each pole. At the end of prophase 1 and during metaphase 1, the chiasmata that hold the bivalent together slip away from the centromost toward the ends of the chromosome. This phenomenon is called terminalization, okay? Slipping to the ends. Reflects the growing repulsion between the numbers of each chromosome field. And we've got anaphase 1, which is already, you can see here, switch crossed over segments of the chromosomes. The pair chromosomes separate from each other definitely. And the separation is called chromosome. Hello, Hussein. Hussein. Okay, I will turn off your mic then. Okay, I have begun this lecture at first o'clock, so you can see the beginning when this will be a pause.
Okay, no problem.
Okay.
I will watch the recorded lecture.
Yes, yes. This is
Okay,
fundamentals of medicine. Goodbye. Have a nice day, Hussein. Bye bye. Nice to see you and hear you. Okay, I will go on now. So we were talking about anaphase when our beloved Hussein joined us. So as the separating chromosomes gather at the opposite poles, the first meiotic division comes to an end. Okay, this is the end of the meiosis 1, anaphase 1, and we've got the different poles of the. So we've got the chromatids chromosomes that always have crossed over, exchange their parts within each other. Okay, and the telophase 1 begins. Okay, the spindle apparatus is disassembled. The daughter cells are separated from each other by membranes. The chromosomes are decondensated, and the nucleus is formed around the chromosome in each daughter cell. Okay. In some species, chromosome decondensation is incomplete. The daughter nuclei do not form, and the daughter cells proceed immediately into the second meiotic division. Okay, the cells produced by meiosis 1 contain haploid, or half of the number of the chromosomes than the mother cell owned. However, each chromosome consists of 2 sister chromatids, which may not be genetically identical, but they might have exchanged material with their partners during prophase 1, okay? Crossing over, remember? So, and the cytokinesis begins. Cytokinesis, as I said, the separation of two cells. Here, after telophase 1, the cell divides into two haploid daughter cells and enters meiotic interphase. Okay, interphase. It is I would say gap between the meiosis 1 and 2. The spermatogenesis, the cytoplasm is more or less equally divided between the two daughters, but in oogenesis, one product receives, or secondary oocyte in this case, receives almost all the cytoplasm, and the other product becomes the first polar body, okay? Just one is the fully functioning one. And the contrast to mitosis, interphase is brief and meiosis 2 begins directly, okay? The notable point that distinguishes meiotic and mitotic interphase is that there is no S phase. We've got no DNA synthesis. As I said in the beginning, DNA synthesis is in between mitosis, right? And during meiosis in the interphase, we've got no S phase, okay? No the DNA synthesis. As the meiosis 2 begins, the second meiotic division is similar to the first one or to an ordinary mitotic, except that the chromosome number of the cell entering meiosis 2 is haploid. So already we've got haploid cells entering the meiosis 2. It means they will be reduced, okay? The end result is that the two daughter cells from meiosis 1 divide to form 4 haploid cells, okay? Each of them contains just 23 or half of the chromosomes that the mother contained, okay? Because of crossing over in the meiosis 1, the chromosomes of the resulting gametes are not identical. Just as its maternal and paternal chromosome in the homologous pair segregates randomly into a daughter cell in meiosis 1, segregation of the different parental and maternal, paternal, and maternal alleles of each gene also take place during meiosis. And this is the last part. Whether the alleles segregate during the first or the second meiotic division, that depends on whether they have been involved in a crossover event during meiosis 1. Okay, that was all I wanted. This is the graphical representation of meiosis 2. You can say that the meiosis 2 starts with the already haploid one. So as a result, we've got 4 haploid cells. Okay, prophase, metaphase, anaphase, telophase, all the same, just like mitosis. Okay, please use your brain, and that was it. Okay, Hussein, I will send you the PPT after you get the uh, save changes. So with the lecture, you will get the PPT and the books. I hope you got the books. We've got 2 choices here: Principles of Genetics or Thomson Genetics. Okay, you can use either of them. I usually combine both of them in my PPTs, but you can choose one of them. Okay. Okay, hope you will find this subject as well as important. I will try my best to make everything clear, okay? If you have any questions, please remember them. Don't forget, write them down and let's discuss them next lecture, okay? I'm not sure whether it's going to be next Saturday or maybe next Saturday, but we're for sure going to meet each other. And see you by then, okay? Bye bye. Have a nice weekend. Have a nice day. And stay healthy. Bye.